Mutations. What are mutations? Mutations are changes in the nucleotide sequence of DNA. So here's a double helix of DNA, and this might have been cytosine, and now it's changed to adenine, maybe. Mutations may occur in somatic cells. Somatic cells are body cells. So your skin, your liver, everything except your ovaries and your testes. These are not passed on to offspring. They can affect you, but they won't affect your babies. However, a mutation can also occur in gametes, the eggs in the sperm, the germ cells that cause the eggs in the sperm, and these are passed on to offspring. So that will affect the next generation. So to summarize that part, somatic mutations occur in non-germline tissues, and they cannot be inherited. Might happen for breast cancer, for example. Germline mutations happen in the ovaries and the testes, and they're present in the egg and the sperm, and that can be inherited. Mutations happen regularly. When your DNA polymerase is copying DNA, sometimes it messes up, and it's not always because of a chemical or ultraviolet radiation. Sometimes it just happens. But mutations can also be increased by chemicals and ultraviolet light, cigarette smoke. Many mutations are repaired by proofreading enzymes, so by enzymes. Are mutations helpful or harmful? And the answer there is yes. Mutations are all these things. So some mutations may improve an organism's survival. These are beneficial mutations. So if there were some mutation that could improve the eyesight of a cheetah or make it run a little bit faster or jump a little bit quicker, that would be a beneficial mutation. If there were a mutation in clownfish that allowed them to, I don't know, maybe be even more able to live with the sea anemone, maybe not, be totally resistant to its stings or whatever. Maybe make it hide better, something like that. That would be a beneficial mutation. This frog has had lots of beneficial mutations in its past to make it look, to make it camouflage, so it looks just like a rock. And the peacock has had lots of beneficial mutations along the way to make it beautiful so that the peahens think, oh, you're gorgeous, and then this one will be more able to get its genes into the next generation. Of course, mutations aren't always beneficial. Some mutations are neutral. They don't affect the organism at all. That can happen if the mutation happens in some non-coding part of DNA. Or, check this out. You saw this in the last unit. The genetic code is redundant. So, for example, UUU, this is the mRNA now, codes for phenylalanine, but so does UUC. So let's take a look at what that would do. If your DNA is AAA, then your RNA will be UUU, and your amino acid will be phenylalanine. If there's a mutation here, and you get AAG instead, your RNA will be UUC, and look at that, you'll still end up with the same amino acid. So sometimes a mutation really does not affect the gene, the protein that's made at all. And then there are some mutations that are bad, harmful. So there are some types of skin cancers and leukemia that can result from somatic mutations, mutations after the organism has started growing. But mutations in germlines can be passed on to offspring, and some of those are bad too. So let's look at types of mutations. There are gene mutations, and that's what we're going to spend almost all of our time on. So here's a mutation. This is supposed to be TGC, and it got changed to TAC. So that's one particular point mutation. So a change in the nucleotide sequence of a gene is a gene mutation. It might involve only a single nucleotide, and that might be due to copying errors. So you put the wrong nucleotide in. Could be due to chemicals, could be due to viruses. Here are the types of gene mutations. Point mutations, which mean it's just one nucleotide, one base that's messed up. Could be because you substitute the wrong one. Could be that you insert one or delete one. A frameshift mutation will happen with an insertion or a deletion. It will shift the whole reading frame over, so I'll show you what that means. Nonsense and missense also can happen when the mutation happens in a certain place. A mutation doesn't have to be just a point mutation, though. It could involve 2 or 3 or 4 or 5 or, a dozen nucleotide bases. So we're going to look at these. So we'll start with point mutations. This is a change of a single nucleotide, and that happens sometimes and causes pretty dramatic results sometimes. So it includes either deleting that one or inserting a different one, or substituting, changing a C to a G. Just if it's a point mutation, it would just be one nucleotide. Of course, you could delete more than one, or insert more than one, or substitute more than one. So here's one example. Sickle cell disease is the result of one nucleotide substitution. So these are red blood cells, and these three are normal, and that one's sickle. And so inside a red blood cell, you have hemoglobin. And hemoglobin is that really important protein that carries oxygen. So it's got this globby structure to it. So that's normal hemoglobin. Sickle hemoglobin will look like that until it becomes oxygen deprived, in which time it will form these spikes. And so that's why you end up getting a red blood cell that looks like this kind of weird shape, because the hemoglobin has it's not globular anymore, it's fibrous. So this occurs in the hemoglobin gene. So let's take a look at it. This is a sequence in a normal adult hemoglobin. So they're not showing you 1 and 2 or anything after amino acid 9. But here's 3. So CTG in the DNA will code for leucine, ACT for threonine, etc., etc. And here's the normal one, GAG codes for glutamate. Here's the sequence in the sickle cell anemia gene. It's just this one amino acid that's, it's just this one nucleotide that's different. So you can see GAG is still the same, AAG, TCT, that's all the same. So this is called a substitution when that happens. You didn't lose an amino acid, you still have the same number of, you didn't lose a nucleotide, you still have the same number of them. You didn't delete one of them, which means losing them. You didn't add another one in, you substituted. Instead of A, now you've got T. And that codes for a different amino acid. And so when the normal hemoglobin folds the way it does, this one doesn't do that. So it folds a different way, and so you have a different protein. A frameshift mutation is a little bit different from that. This is when you insert or delete one or more nucleotides. So this might be a point mutation, might not. But it changes the reading frame, like changing a sentence. So I'll show it to you first in English, then I'll show it to you in DNA. So here would be one in English. The fat cat ate the wee rat. Notice that we picked a sentence here that has all 3-letter words, which is the way DNA works. A frameshift substitution would be losing a T, and so I think this is the T we lose. So the first 2 words are the same, but the next word is not if you're deleting this. So it would be CAA for the reading frame, because remember you need 3 amino acids. You need 3 nucleotides to code for an amino acid. And so what this does is mess up the whole reading frame. So the next word is TET, and the next word is HEW, and the next word is EER, and so on. So a frameshift mutation will mess up the entire thing after the mutation, whereas a substitution only will typically mess up one codon to mess up one amino acid. The amino acid sequence is changed for the rest of the gene. So here's an example. Here's DNA, TAC, CTT, etc. Here's the DNA. Here's the RNA. So T is complementary to A to U, C to G, etc. And so these are if you get your handy-dandy chart out, you can see the amino acids that are coded for by this, and here they are. And so here's the mutation. Let's see if we can spot it. Here it is. So I've got TTC. Okay, so what happened here is a deletion of this, because look, TTC, got that. GTG is that one. CA, and then that must have been the one after. So this is a deletion, which is causing a frame shift. Because instead of having one codon different, now all the codons after are different. So this is the transcription into mRNA, and then the translation into amino acids. The first one's the same, the second one is different, the third one is different, the fourth one is different. So it's really shifted the entire reading frame. Now the protein doesn't make sense anymore. That's a frame shift mutation. So let's take a look at the animation. The original one is this. Substitution is when you change one for another. Insertion is when you add a new one, changes the reading frame. Deletion, you remove one, it changes the reading frame. Inversion, you just switch spots. I hadn't gone over that yet. So let's look at this again. Original is here. Substitution, you change G for C. Insertion, you're adding one in. Deletion, you take one out. Inversion, you switch a couple. Missense mutations. That occurs when a mutation causes a different amino acid to be used. So here's the same one we were talking about before. Here, instead of deleting one, you just changed it to a G from a T. And so the whole rest of it is okay. And so now you have a different amino acid than you had before. It didn't change the whole reading frame because it was just a substitution right here. But that would be a missense mutation. A nonsense mutation is interesting. This occurs when a stop codon is produced before the end of a gene. So here's the gene and the mRNA, and here's the amino acid sequence. And this might go on even longer. Here you change a C to a T, and that happens to code for stop. And so now the protein's much smaller than it would have been, and it can cause some pretty major problems. Neutral mutations. Some mutations do not occur in coding regions of DNA. Other mutations occur in a coding region of DNA but still code for the same amino acid. So I showed you that one already. So, for example, this is the DNA, here's the RNA. If you change this T to a C, it turns out that you actually get the same amino acid still. Chromosomal mutations. So those were all gene mutations. A chromosomal mutation is when you change the structure of a chromosome. You gain or lose a part of it, or you shift it over to a different one. And we covered that in meiosis, so I'm not going to cover that again. We covered that in abnormal meiosis. And that's it.